Hi, everybody. I'd love to introduce Kate Adamala from University of Minnesota, and she's been trying to, well, synthesize cells to, like actually drive them like, well, computers, if I may say so. Welcome, Kate.
Thank you. Thank you so much. Yes, what we're trying to do is make fake cells and have them behave a little bit more predictably than natural cells in order to do biocomputing. And so I want to start with just a very brief kind of a framing of what are we talking about, because a lot of the time when you start talking about biocomputing, you can spend half an hour talking to someone, and at the end you realize that each of you actually means something else when you say biocomputing. So you've been having conversations at each other rather than with each other because you don't agree on definitions. That happened to me many times when I think of biocomputing as something very specific biological processes. And then someone else thinks of it as something completely different. So let me start by defining what I think biocomputing is. And whenever I'll say Boolean logic gate operations in biological systems in this talk, this is exactly what I mean. So what I mean is you take a piece of genetic information and you make it produce some kind of a signal. And this is the very simplest, the lamest, but also the simplest possible definition of what a single biological logic gate is. So when you think about it, every instance of a protein expression in biology is a logic gate, because you have some sort of an input that tells you whether that protein needs to be expressed or not. And then you have an output, which is the protein signal. And so when I say biocomputing, that's what I mean, is taking some sort of an input and making a biological output, most of the time a protein, out of it. And so this is obviously useless the way it's drawn, because what can you do with a single logic gate? But this is the chassis that we're building on. And this is the kind of a chassis that the field has been using to build more complex systems. So once you define what a single logic gate is, then you can start stacking them. You can start building more complex systems, more complex networks, all based on this very simple principle, is that you have some sort of a chemical input, then biochemistry happens, and out comes some sort of an output, a lot of the time fluorescent or small molecule, but some sort of an output that tells you the state of your gate. And notice that I'm talking in a very abstract terms. We're not doing anything useful here. It's not computation that is going to be a counter or anything. This is so early that this is not yet useful. So don't ask me what is it good for, as in what can we actually do with it? Nothing. This is proof of principle still. This is still very early work, but this is the principle that we've been using, which is, let's pretend biological systems are logic gates. Okay, so this is the framing that got this field going, all the way back 15, 20 years ago when Chris Voigt first started putting biological logic gates into cells. And the way people have been doing it is using that principle of, let's make a protein expression system that responds to a specific input and produces a defined output. Let's put it into a cell. And that obviously presents a problem, because natural cells are incredibly complex. They first order of business of any natural cell is to stay alive, to replicate, make more copies of itself. And then once it replicates, it wants to eat, it wants to have metabolism, all of those processes that we still don't understand because we don't have a complete chemical list of ingredients of any living cell. So imagine we're trying to build those logic gates, we're trying to build those computational systems, and then we bury them deep into this gray goo, into this soft matter physics that no one really understands. And that's been obviously a problem. The reason we still don't have a functioning biological computer is because that goo is soft and does what it wants rather than what we want it to do. And that's basically that's where my field comes in, and that's how I got into this. I'm not a computational biologist or by training. I got into this because I'm a chemist who builds cell-like systems that are not really natural cells. So basically, our approach to this problem is that instead of trying to build those biological computations into a complex living cell that we don't understand, what we want to do is we want to simplify that chassis. We want to build this computational system into a chassis that's a little simpler. And that's that synthetic cell. And I'll tell you in a second what a synthetic cell really is. But what we want to do is we want to take those whatever logic gates you want to build and still rely on biology. So still take advantage of biological signal amplification, of biological principles of design of those genetic circuits, but we want to put it in a chassis that's much simpler. So how do we actually do that? How do we get there? So there's kind of two different ways in which people were trying to do this biological computation, the computation with biological systems. And one is what I showed you couple slides earlier, is using living cells. So encode those genetic circuits into a complex living cell. And then the other way of doing it is to do it completely in vitro, so in dead cells or outside of the cells. So just take very few purified molecules that we understand, put them together and try to do your computations there. So one obvious advantage is that if you do it in vitro, it's very programmable. You can predict what happens. In live cells, the cells do what they want, so your gates are going to be very leaky. Unfortunately, when you do it in vitro, you don't have signal amplification, because that's what biology gives you. That's why we want to use biology. Is if you have a logic gate in biological system, you're going to have signal amplification at many levels. If you do it in just a dead in vitro system, none of those signal amplification mechanisms just don't exist there. In vitro doesn't replicate. So whenever you want to make a biological computer, a logic gate system, you have to build it from scratch. However, it actually works in vitro. If you build it, you tell it what to do, it's going to do what you tell it to do, because it doesn't have a mind of its own. Living cells do. They evolve, they change, they accumulate mutations over time. In living cells, you're kind of stuck to what you can use. Nature is not very creative when it comes to the use of molecules. It's only 22 amino acids, it's only 5 nucleobases. It's really difficult to expand that chemical chassis. While in vitro, you can do that. You can actually put whatever molecules you want. And last but not least, living cells, because they're living, they can also get dead. If something is done in vitro, you can store it for extended periods of time. You can freeze dry it and store it at room temperature for a long time. So these are kind of design constraints that have been applied, that apply to both in vitro biocomputing and biocomputing in living cells. So I told you that our field kind of has a different approach to this. And so what we do is we're trying to do this in synthetic cells. And so synthetic cells kind of offer you advantages of both worlds. This is obviously a little bit of cherry picked, as we usually do for talks. This is highlighting the advantages. The biggest disadvantage of synthetic cells is that they still don't self-replicate. So you have to build them every time, which is actually a bug and a feature. It's a bug because you have to make it every time, but it's a feature because it increases the biosafety and biosecurity of those technologies. They're not going to replicate, go on their own. But generally, synthetic cells offer you that huge level of flexibility. So you can build something in a synthetic cell using arbitrary components. You can design it to specs. It's not going to evolve. It's not going to go do whatever you want, whatever it wants. It's going to stay on program. Okay, so I've been talking for like 5 minutes now, and I still haven't told you what a synthetic cell is. And that's on purpose, because no one really knows. The field is young enough that we don't have a good definition. To me, a definition of a synthetic cell is something that doesn't come from an unbroken line of life that's been on Earth for the last almost 4 billion years. Because when you think about it, everything alive today comes from unbroken line of other living organisms. You and your dog and your gut microbiome and the plant on your window, everything living right now has been alive since the origin of life on Earth. What we want to do with synthetic cells is we want to break that chain. We want to use biological parts, but we want them to be dead. And then we want them to put them together and eventually have them again become living. And again, that's not a definition. I realize that. So if I'm forced to give a definition, I would say a synthetic cell is some sort of a biochemical system that has emergent properties. So it's more than a sum of its parts, but it's still controllable. And that controllability is really what drives the field. I'm a chemical engineer by training. So when I started doing biology, I was really almost disgusted by how uncontrollable it is. You put things together and they just go on their own. They evolve. There's this whole epigenetics thing happening. You put molecules in there, you don't know what comes out. I want my systems to do what they promised me to do at the beginning. And that's that controllability part that we have in synthetic cells that we don't have in natural cells. And some of the things that we can control in synthetic cells are things that you cannot control in a living system. Things like the size of a cell. In synthetic cells, we can control the size by couple orders of magnitude. You cannot do that with a normal living cell. We can control the very chemistry, the very nature of the chemistry. So we can control how we make proteins. We don't even have to make proteins. We can control the chemistry of every part of the metabolism. And so that controllability is really what gives us this engineering flexibility. Okay, I still haven't told you what I mean when I say a synthetic cell. So finally, this is what I mean when I say a synthetic cell. I mean a liposomal bioreactor that makes proteins. This is the very simple definition that my lab uses because we're liposome people. So we think a cell should have a membrane. That's actually where a definition, the word cell, came from the fact that a guy who invented a microscope looked at first samples under the very first microscope, and they kind of looked like those cells that monks live in, the little square compartments, and that's why he called them cells. And cells should have compartments. That's kind of what we believe. And for evolutionary purposes, for application purposes, we want a membrane. And now what goes inside that membrane is really the fun part. So we really we focus on building what's inside it. So when we make synthetic cells, they all have a membrane most of the time. And that membrane is not different than the membrane of your own cells. We use the same lipids that your own cells use. We also use cholesterol. We love cholesterol. Everyone's scared of cholesterol because the doctor tells you it's bad for you. Cholesterol is actually not bad for your membranes. Cholesterol is awesome for your membranes. It makes it fluid. It's only if you have too much of it and cells don't know what to do with it, they accumulate it where it doesn't belong and it clogs your arteries. But cholesterol in biological membranes is actually good. So we use a ton of it. We sometimes also use simpler fatty acids when we want to make simpler membranes. Now, I have to show you the next slide because we also do membraneless microfluidics. A postdoc in my lab, Theo, would be very upset if I don't mention microfluidics. We do have microfluidic systems that don't use membranes, but for most applications, we do want a proper membrane. Sometimes you just want to make a lot of pretty movies of identically looking bioreactors, and that's why we use microfluidics. But okay, compartment, that's the last word I said on a compartment. What we really focus on is what goes inside that compartment. So what do we put inside a synthetic cell? And what we do put inside is called a cell-free protein expression system. So it's cell-free because by the time we get our hands on it, there's no cells anymore. It still comes from natural biology. So when I told you we want to move away from the constraints of natural biology, this is still aspirational. We haven't yet been able to completely make a protein or translate a biological information without any biological parts. So most of the time what we do is we're cheating. We're stealing biological parts from a cell, most of the time bacteria because they're the cheapest and easiest to grow. We extract all the components from a bacteria that the bacteria uses to make proteins, and then we modify those components. We change the chemistry of it, and we put it back together in a test tube or in a liposome or in a microfluidic system to reconstitute that translation system. And that's our synthetic cell. Now, what is it good for? And why do we do it? For many different reasons. This is just a kind of a summary slide that shows you what a lot of different areas a lot of different reasons why people are excited about this field. There are practical applications. We're developing tools, we're making new drugs. This is very early technology, but we already have evidence that shows that synthetic cell-based drugs can shrink tumors in a mouse tumor model. This is work from Avi Schroeder at Technion. So it is emerging technology, but we already have evidence that shows this one day might be good for something. So this is kind of a getting towards applications. We're developing a lot of different tools. Many of you probably heard about nanopore sequencing. You guys work with big data, and a lot of big data right now comes from nanopore sequencing. That technology have been developed in synthetic cell engineering field, because the first alpha chemolysin channel that gave rise to the nanopore sequencing was developed by Dave Dimmer when he was developing the first pores for a synthetic cell. So that shows you this interplay between tool development in building chemical systems that resemble biology and actual studying biological systems. Then it also is metabolic engineering. There's safety security applications. There's the fun esoteric applications like understanding the origin of life, because, as Feynman said, what I cannot build, I cannot understand. You cannot claim to understand how life started on Earth or how life could start elsewhere in a solar system unless you can actually build a living system from scratch. So that's what we're trying to do. And then the reason we're all here, which is biocomputing. So this kind of a corner right here is what we're really going to be focusing on today, is how can you do computation with biology in a way that actually is high fidelity, makes sense, and allows you a lot of flexibility, more flexibility than natural systems. Okay, so a little highlight for nerds like me. The reason why I'm really attracted to this field is all the possibilities that are not possible with natural cells. So the one nice thing about synthetic cells is if I'm building a synthetic cell, I can give you a complete chemical list of ingredients of that cell. And if I can give you a list of ingredients, that means I can digitize that list and send it somewhere without the physical material transfer. And that's impossible in biology. When you make a strain in biology, if you want your collaborator to work on that strain, you have to ship him that strain. I working on synthetic cells can send you electronically information about a full ingredient list to make a synthetic cell, assuming we agreed on the list of the building blocks earlier and you have the same building blocks. And so that enables what we jokingly call teleportation of biology. It enables sending information about building of biological systems over great distances. And that's, obviously, if I have a collaborator in the Netherlands, then I can send him an email, or I can just send FedEx him a sample. But there are some situations where FedEx doesn't deliver, and this drives the applications of that field for things like astropharmacy. Once Elon Musk goes to Mars, we cannot predict in advance what healthcare needs he's going to have, what nutrient needs are going to emerge. But we can build a biomanufacturing platform that we can then use on demand and program remotely. And that's the kind of a fun, nerdy part of that field that we're really working on. But that doesn't apply to biocomputing, so I'm not going to focus on that too much. I just want to highlight that this field is good for many different things, some of them crazy, but actually possible. Okay, so I told you we want to make cells from scratch. We want to make cells from non-living components, and no one has ever done that. So half of you are probably now thinking I'm full of it and this is never going to happen. Okay, so I want to show you a quote from an article published in the New York Times. Okay, so New York Times, a very respected publication, published this article in October of 1903, and it was an article on the feasibility of building a flying machine heavier than air. So it's 1903, we have dirigibles, blimps, we have balloons. So we know a human can build a flying machine, but they all have to be lighter than air. That's the common knowledge. We all know that to fly in a mechanical device, that device has to be heavier than the surrounding air. That totally makes sense. And so they wrote this article saying a flying machine heavier than air is practically impossible. You might as well not waste your time doing it. And then 2 months later, the Wright brothers flew their first Wright flyer. The reason I love showing it is because, I'm coming from the field that claims that we soon will be able to put together biological molecules into a living cell from non-living components. And that sounds absolutely crazy, and people tell me this is impossible. And my reply to that is, anytime you tell scientists and engineers that something is impossible, we're just going to go out and do it out of spite, because you tell us it's impossible, we're going to go and do it. And so that just kind of shows you that the field, even though we're still early, it's really getting there. Nothing is impossible unless you violate laws of physics. So this is where we are.
It's a good time, if I may ask, in some ways, this is pretty clear that some variants of this is possible. Let's do microfluidics. It sounds like what you're focusing on is some biochemical, mostly biochemical reactions in some kind of container so it doesn't leak. So yes. A microfluidic chamber mostly does that, plus
Yep.
some amplification reactions to make sure whatever you produce get amplified a lot. Is that fair as a core capability?
Yes, that's a very good description.
Okay, so then where does that stop? If you just have a giant array of microfluidic reactors, is that good enough?
It's good enough for me. Some people, some hardcore evolutionary biologists would tell you that it's not good enough because you don't have membrane. Some people really think membrane is a hallmark of life. Even though I'm a liposome person, I love membranes. I don't think it's a hallmark of life. As long as you have a microfluidic chamber, any way to compartmentalize your reactions, that's good enough for me. That evolution can happen, biochemistry can happen. I'm happy.
Okay, so then I'll do the completely other way. What's so hard about that? We have microfluidic reactors, we have chemical reactions. You can shove any reaction you want in there. What are the primary challenges before it becomes something useful for computation, for manufacturing?
So useful that it already is. We already have a lot of application that's useful. It's not yet living, and that's the problem. A lot of people think that in order to really call it a synthetic cell, it has to be alive, and that obviously we're crossing into the definition of life areas, which is a murky, muddy area. No one knows what life is. But in order for it to be alive, at least for me, it has to self-replicate. And our systems do not self-replicate right now. So that's where we're going. We really want it to start self-replicating.
So why? Because you gave a bunch of applications. What applications are satisfied by self-replication as opposed to assisted replication, perhaps to control numbers?
Yeah. So for practical applications, you don't need self-replication. In fact, it's better if you don't have self-replication because it's much safer. The technology is much safer if it doesn't self-replicate because then it cannot run uncontrollably. So that's why this field, that's where all the money in our field is coming from, is from those practical applications in biotech. For example, a lot of metabolic engineering companies right now already uses cell-free systems for prototyping. You don't need self-replication for that. You don't want self-replication for that. Same for medical applications. If you're going to inject it into a patient, you would rather it not self-replicate. The reason we want to self-replicate is because we're self-important and we want to say we made life. We want to say we recreated the process of the origin of life. So for that, we really need self-replication. We want evolvability in order for it to really say we build an orthogonal tree of life. So these are all foundational research reasons.
I see.
And there is some biomanufacturing applications for which you want self-replication, because if you want to build a bioreactor that produces molecules that are commercially viable, you want that bioreactor to be able to self-renew. So that self-replication is important for that. If you really want to bring the cost down, because right now this is a ridiculously expensive field because we need to build those biochemical systems from scratch. If they were willing to self-replicate, then I could build it once and then keep making more of itself. So the cost would go down. And also for biocomputing, this is actually one of the areas where we do need self-replication, because if you want a biocomputer, you want it to persist for a long period of time. And in order to do that, it probably should make copies of itself. It should renew itself at least for a while.
But presumably you want very controllable replication for safety, obviously. But I'm thinking, like, how does this thing as an application different from, let's say, algae? They're a bunch of cells of various sorts. You want them to reproduce so you have a stable population, but they also crash populations repeatedly, and because they're very unpredictable, parasites and so on, they destroy them.
Yes.
So how can you have highly controllable replication? Because it sounds like that's really what you want.
We really want controllable replication, and that's why we're building replication from scratch. So we're not relying on natural mechanisms that make cells replicate, but instead we're kind of trying to hack it. We're trying to build a replication system that doesn't rely on the natural mechanisms, so we can build the control on the ground up. So for example, we're actually just submitted a paper where we're showing one possible mechanism for the replication of those non-living cells, and that mechanism doesn't rely on cytoskeleton, doesn't rely on all the natural elements of the cell replication that can make it uncontrollable. Instead, because it relies on very specific proteins, we have more ways to control it. But it has not actually been done in published literature yet, so it's all speculative.
Right, that makes sense. All right, cool, thank you.
All right. So any more questions, please just stop me.
Mhm.
Okay, so now one specific project out of my lab, the reason you guys wanted me here, biocomputing. So we had a we developed this platform called Trumpet for showing the utility of using synthetic cells in biocomputing. So why did we call it Trumpet? Because the first biocomputing platform ever developed was called Cello. It came out of Chris Voigt's lab, and it just sort of became a joke in the field that a biocomputing platform should be named after a musical instrument. So when we developed our platform, we worked really hard. This is one of the craziest backronyms that I ever had to come up with. We wanted to name ours after some sort of an instrument, and the lead grad student on this project plays trumpet, and she really wanted to name it after trumpet, so we made it work, but it's a mouthful. Okay, so what is a trumpet? A trumpet is a way to perform Boolean logic gate operations in a in vitro system. So what it does is it uses a transcription system. So transcription is when you make a functional fluorescent piece of RNA out of a DNA template. So every trumpet gate starts with a DNA template, and then a bunch of enzymatic reactions happen, and out comes a fluorescent piece of RNA. So it's fluorescent because we want to be able to read the signal from that logic gate. And I'll show you an example of a NAND gate to show you how that trumpet gate works. So a trumpet gate starts with a single-stranded DNA template. So it's just a string of DNA, a single-stranded string of DNA. It's about 150 to 200 bases, not base pairs. It's a single-stranded DNA. So it's a short, in IDT terminology, it's an ultramer. And that piece of DNA has three regions. There is a promoter for an RNA polymerase. There is the actual logic gate area. So this is where we do our operations. And then there is this readout area. So this is where the fluorescent aptamer is encoded. So this is what eventually will tell you the state of the gate, whether it's on or off. And so in case of a NAND gate, what happens when we want to do a NAND gate operation here? So the first thing we do is we can add a restriction enzyme. So these are the normal restriction enzymes. If you ever took molecular biology, you probably heard about restriction enzymes. These are the favorite enzymes of molecular biologists. These are the enzymes that we use to cut DNA when we do cloning. So that restriction enzyme gets added to the sample. And then we also get what we call input oligos. So these are the ways to program our gate. Short, single-stranded DNA oligos that we add to the gate to perform operations. And so if we have our NAND gate template and we add two input oligos to our gate, what happens is those input oligos hybridize or bind to that logic gate area of our gate template. So now what you have is you have a double-stranded DNA because you added two input oligos that completely covered that logic gate area. And so now suddenly that logic gate, that computation area becomes double-stranded DNA. And what happens when it becomes double-stranded DNA is that the restriction enzyme now becomes active, because the restriction enzymes that we're using are very particular about what they cut. They will only cut a double-stranded DNA. So they're sequence specific. They only cut specific sequence, but they will only cut that sequence if that sequence is double-stranded. So if they're faced with a single-stranded template, they do nothing. Only if the template becomes double-stranded, the restriction enzyme will cut it. And now what happens is the restriction enzyme cuts, and it separates that promoter region from the aptamer region, because the cut happens here, right when that logic gate region is. And now, if you try to do a transcription, so if you try to add an RNA polymerase to your system, it will not produce a fluorescent signal because the RNA polymerase needs to start at the promoter and continue to that fluorescent aptamer region. And because you cut your template right here, now the RNA polymerase will bind to the promoter, but then falls off because it has been cut here. So there's nothing to produce signal. So in presence of two inputs, you end up with an output that is 0 because there's no fluorescence. So now if we don't add any inputs, or we add only one input, now the restriction enzyme doesn't know what to do with it because it's partially or fully single-stranded. It will not cut. You will end up with your intact template producing a fluorescent signal. And if you think about it, that is a truth table for an AND gate. So in the absence of inputs, or if there's only one input, you end up with a signal. And in the presence of two inputs, you end up with a output that is 0. In our case, 0 is no fluorescence. And so you basically encoded an AND gate in a trumpet system. It takes about 2 hours to do that entire reaction. And does it work? Well, I'm obviously showing it, so it has to work. It does work. That's just one example of a type of a fluorescent signal that we get out of a NAND gate in the trumpet system. So in case of 0 inputs or only one input, we get signal. In case of both inputs, we get no signal. And is it orthogonal? We tested, these are 2 examples of mismatches. We actually tested gazillion of them. There was actually an undergrad project for at one point was to keep testing mismatched templates with mismatched signal oligos over and over again. And we found out that it's very orthogonal. Basically, if you have mismatches, you will not get a signal. So it's very high fidelity.
So actually, this sounds like it's more than binary NAND gate, because you can be highly selective about what the two inputs are, right?
Yes, yes. And we can have multiple, and we've done that too. We have multiple NAND gate in parallel in the same reaction.
Right. And now how much, now I'm thinking, because you can do many possible sequences for the 2 bits. In fact, you can have more than two inputs, and they can have more than 2 values. Can you have any wildcards where you can have a set of possible matches, bind to a particular subsegment?
In theory, yes. We've never tested that, but it's possible to use wobble base pairing. So DNA base pairing that's ambiguous and build those wildcards. We haven't done it yet, but it's possible.
Okay, and then the other one is your output, your readout section is a fluorescent thing. So you can just read it from an optical sensor. But that's, that pushes you out of the biological system. Presumably you want a chemical to be coming out that feeds into the next gate. Is that the real application?
Yes, and we've shown that. This is the next step. Okay. So this is how you layer the gates. So what I've shown you before was just an example of a single NAND gate. This is how you layer the gates. We have a solid state support. In our case, that was just a magnetic bead. It could also be a microfluidic system. On that support, we immobilize a piece of DNA with a biotin anchor. On that piece of DNA, we immobilize our gate template. So this is the same gate template, the same type of a gate template that I showed you before, except at the end, instead of a fluorescent aptamer, we have what we now call a signal oligo. So this is a short oligonucleotide that can be used to program the next generation gate. So instead of producing a fluorescent signal out of that gate, the output is that signal oligo. And so now if we have 2 inputs, and then we also have this so-called release oligo, which is going to become important in a moment. But this is the general design of, again, an AND gate on solid support. So if we have 0 inputs, we have no cut. 0 inputs or 1 input, we have no cut. Now we're adding our signal release oligo, and that signal release oligo creates a new double-stranded area. Because remember, I said restriction enzymes that we use only cut if it's double-stranded. So if the gate has 0 or 1 inputs, now we have the signal release oligo that cuts the output and creates this signal oligo that can go to the next gate. Now, if we have 1 or 2 inputs, it cuts. So our gate is now cut in half, and it floats away. We wash it. Now, if you add your signal release oligo, it has nothing to bind to because its binding partner was already washed away. And that's why there is no output out of that gate. And so what goes to the next gate is 0. So that's the fluorescent-free way of generating 1 or 0 output. And we've shown, for the sake of time, I'm not going to go through a ton more data, but we've shown that you can layer those gates. We've shown that you can have all the universal types of gates encoded in the trumpet systems. We've shown NOR, OR, NOT, and NAND gates. That's a mouthful. We also developed a cloning system to bring the cost of the whole thing down. We showed that it's orthogonal, and it seems like every project must have a website these days. So we have a website where you can go design your gates. So again, this is proof of principle. We just showed that you can encode very simple gates. We're working with a collaborator right now to make an adder. So again, it's still going to be a proof of principle adder, but it's possible to encode an adder in Trumpet. At least preliminary data shows that it is. Okay, so it works. It's possible to do those layered biocomputing operations in a very simple in vitro system. And I'm going back to this slide of potential applications, because it's not just good for biocomputing. It's good because a logic gate is a way to control a biological system. So when you think about it, it's also possible to control, for example, gene expression. So you can control how your cells behave. You can build those systems to control protein expression, to control, for example, drug development, if you want to use it as a drug making platform. And now I want to spend the last couple minutes showing you something that's not biocomputing related from the start, but it's really cool and can interface with that system. So the idea is that synthetic cells could be used to make drugs during spaceflight. And that was an idea that people had for quite a while, the so-called astropharmacy, where you use those very stable biological systems to make drugs in space during spaceflight conditions. And how do you control that? How do you build precise control into those systems? And that's where the cross interaction with biocomputing comes in. We can use biocomputing systems, so those logic gates, to program behavior of those on-demand biomanufacturing systems. And now the question is, it sounds great on paper. Does it actually work under spaceflight conditions? So there was this project by people that are really interested in making astropharmacy applications, in making biologically active drugs under spaceflight conditions. And we were very happy to be part of that team that set out to test whether those systems work under those conditions. So we built 2 different reactions. So what's shown here is a transcription of a fluorescent or a functional RNA. This is the very same reaction that I showed you a few slides ago in a trumpet system. So this is the exact same RNA aptamer called broccoli. RNA aptamers are called after vegetables. So we use the one that's called broccoli. There's corn, there's mango, there are peppers. RNA aptamers are vegetables for some reason. And so here we use the broccoli aptamer, and we used it in a lyophilized form. So we basically freeze-dried it, stored it at room temperature. We also encoded a protein expression system under the same conditions. And we stacked it on a sounding rocket. And here's the video that I want to show you, because I was up at 3 a.m. watching that video. And now I'm watching it to everyone. The cool thing about this video, there are 2 cool things about this video. One is that this is a cell-free protein expression system that's going into orbit, and the reaction happened on orbit, and it worked. So we can make proteins under spaceflight conditions. But the really coolest part of it is that it's my own sample. The sample that I made with my own hands is going into space. I mean, how cool is that? To me, that was incredibly cool. So that was really the most exciting, other than the scientific value of this project. That was the most exciting part for me, is that something that I made with my own hands in my own lab, actually went up into space, and it did work. We did send it. It was a sample return mission, so we had both telemetry and the analysis of the samples after it came back, and now we know that it does work. You can send samples. They survive on tarmac in Virginia in the summer for 4 days, waiting for the launch window, so they can survive huge temperature changes. They can survive pressure changes when they're lyophilized. Then you can rehydrate it, basically add water and make it work again. So back to my favorite summary slide, this is good for a lot of different applications, and it actually works. It works under a lot of different conditions. So last but not least, I want to plug our international Build-A-Cell community. There is literally dozens of us working on synthetic cell engineering. No one gets that reference. There is a show called Arrested Development where one of the main characters used to say there is dozens of us. And every time I use that in the talk, I'm kind of hoping that people like this show because I do, but no one does. And completely pointless reference. There is a lot of people.
I thumbs up on that one.
Yes, thank you so much. I'm not going to paint my face blue, but other than that, that's as close to that reference I can make. There is a lot of labs all over the world that believe that it's possible to build life from scratch, and so we're united in this collaboration called Build-A-Cell. That was actually my first contact with this community. I started my lab 6 years ago, and in the first year of me being in my faculty job, I went to a meeting where people were saying, we should have an international community for our field. Why don't we? And me being a dumb young PI said, I can help doing that. And so I ended up creating that Build-A-Cell community as a very junior PI, which was an absolute dumbest thing one can do. But it did work out. The community still exists. We're growing. We're having now 94 labs. I think it's 96 right now. We have thousands of people that went through the Build-A-Cell system. We have a system. We have meetings. We have meetings twice a year. That's the best part of our community, is that people actually come together twice a year to those Build-A-Cell meetings, and we coordinate research efforts. That's the part that I really like the most, is every 6 months people come together and we have those one-day workshops where we actually work. It's not talking heads, it's actual people sitting down with their computers working. And we develop shared protocols. We develop outreach strategies. We develop ways to share unpublished results and unpublished data. And that's the best part about this community, is that it actually is friendly. And shout out to National Science Foundation for funding us back when they used to fund cool stuff. And I want to say thank you. I'm out of time, so I want to just say thank you to everyone who supported my research. And most of all, thanks to everyone who joined my lab. When I started my lab, I said I want to make life from non-living components. I want to make life from scratch. And I mean, that sounds kind of crazy. And there are people that actually did join my lab and decided, that's not that crazy. I can actually do it with you. So I'm really, really grateful to those people for doing it with me. And thanks to everyone who paid for it. And thank you for listening. And I'm very happy to take questions now.
All right, well, thank you very much. I have a few, and people chime in if you have any additional questions. Just let's say on the computing side, I'm noticing you have these individual gates. So first of all, you showed us a NAND gate. Are you building the other gates out of the NAND gates, or do you have specialized circuits for the other kinds of logic gates?
Both. So we've shown that each of those other types of gates can be directly encoded in Trumpet, but we also showed that you can layer the gates, and we've used a NAND gate to build OR gates and a couple others. So answer is both. We can encode them either directly, or we can layer them to make another gate.
Okay, and then I'm kind of trying to forecast the evolution, the technological evolution of the circuit infrastructure. So I'm noticing the substrate is much wider. It sounds it's natural. It's inconvenient to do electronics with more than binary. I mean, it's hard to differentiate different signals at a high speed. Whereas here, many different matches are pretty much equally easy. But what I'm also noticing is that the speed is much lower. You're not going to get a billion operations a second anytime, well, ever. But what is the actual limit? You're saying 2 hours, but I believe a bunch of cellular interactions happen at millisecond rate. What timescale should we be thinking of?
Yeah. So we've got it down to about 30 minutes. That's still unpublished because it's kind of messy. As you try to push it You give the reagents less time, you start getting more background noise. Biocomputing, I don't think it will ever be a fast technology. If you want fast, you should go quantum. If you want to interface with biology, that's where this actually will be useful. If you want to translate biological signals into digital or the other way around, or if you want something autonomous, so if you want cells that can regenerate, I don't think biocomputing will ever give us a cell phone equivalent. So like a computer that people can carry in their pockets. Maybe I'll be proven wrong, never say never with technology. But right now, it doesn't seem like it's going that way. Speed is really not our strongest feature here, because you can either have speed or fidelity in biological systems. Usually, if you go fast, you're losing fidelity. And so we try to invest in fidelity right now.
Right.
And again, I can be proven wrong. Maybe they'll be made faster one day.
So is this a property of, is it a gene expression usually take this much time to be high fidelity? And you're describing constraints on the gene expression.
Yeah, so this is actually not even expression of genes yet, because we're not making a protein, we're just making a fluorescent aptamer, which is why this is so fast. If you were making a protein, you would have to, it would take at least 4-ish hours for that gate. I think that the problem with speed is that enzymes are slow, really. We think of enzymatic reactions as happening on a millisecond scales, but that's just the reaction. There is this whole process of enzyme has to find its target. A lot of those reactions don't happen even when the active complex gets formed, because it just dissociates and you have to give it another chance. So you basically need to let the biology cook for it to do its thing. And that's sort of an inherent problem to any of those systems. And I don't have a good way of thinking. Maybe someone will engineer around it at some point, but I don't have a good idea right now for what to do.
That makes sense. Actually, like Samuel has a few questions, so thank you. I'll just read them out for everybody. So do you know of any research using AI/ML, especially deep learning, to help you model and simulate these and see how you can help evolve it?
Yes, definitely. People are trying to apply that. I'm actually working with someone right now who wants to build use trumpet data to train a model so we can predict new gates. I don't know anything about how you train your AI dragon, so I can't tell you exactly what algorithms he's using. There are other people in the field using deep learning to predict behavior of cell-free systems in general. So to basically predict how those logic gates or genetic circuits, as they call them, will behave in kind of a free market, like if you just build them and let them run on its own. And they're having quite a bit of luck, as far as I understand it.
And what about things, I'm thinking whole cell models, perhaps miniaturized for this purpose because these are simpler. What are the challenges of modeling a whole cell or a large chunk of one? And is that useful?
Yeah,
I mean,
it's super useful. It's extremely useful. There is a lady called Zan Lutney-Schulten. She runs a whole NSF SRC center that's specifically aiming to do that, to build a whole cell model of a cell. It's incredibly difficult because there's so many moving parts. There is, you're at some point your model becomes so complex that it becomes too computationally expensive to do anything useful with it. But they're chipping away at it. They're making a lot of progress, and I think we'll see that soon.
Okay, excellent. So then Samuel's other question is, what are the healthcare environmental applications of this kind of biocomputing?
Healthcare applications. So one of the applications is in detection and diagnostics. These are little things that you can inject into the bloodstream, and they can do simple computations. The advantage of that is you can have a low level diagnostic events that can be amplified. Actually, the first author of the trumpet paper, she graduated now, and she spun off her own company that's supposed to use the trumpet platform to develop a detection technology for a difficult to detect early metastasis cancer. So there's some types of cancers that are very difficult to detect until they're absolutely advanced enough that you're dead, and or almost dead. And these are very difficult to detect because they're very small, they're very localized and difficult to image. But there are cells from those tumors circulating in a bloodstream. They're just single cells. That's why Theranos was such a big deal before we realized they were full of it. If that technology was real, that would be huge because detecting single cell events in a bloodstream is incredibly difficult. So what Judy's trying to do with her company is use the Trumpet platform to amplify that signal. So basically rely on those logic gate operations and use the presence of this potential cancer cell somewhere in your bloodstream as an input, and then amplify that signal many, many, many times to produce something that's then detectable by traditional diagnostics methods. And that's one application. Down the line, we're hoping to be able to basically remake a CAR T cell. Because right now the problem with CAR T cells is that, yes, they will cure your cancer, but they will also give you leukemia as a side effect, because they're still modified immune cells, and you inject them into a patient, they go do their own thing. We're hoping that we can build those very controlled, non-replicating synthetic cells that rely on those logic gate operations to identify the site of a tumor, release the drug at the site of the tumor, and then disintegrate. So that's still, far off, but the technology is getting there.
Right, but the conditional release of chemicals is literally the gates you have right now.
Yes, and
Okay.
that's one of the areas where we and others in the field are developing applications.
Okay, and then last one from Samuel is the ethics of it. Yeah, like what can go wrong, and how can you keep people responsible for it?
Many things can go wrong, and if they can go wrong, they most likely will. That's the assumption you have to make in any biosafety and biosecurity analysis. And so that's actually another area that I didn't talk about at all, but that's something that I'm doing quite a bit of, is safety and to some extent security analysis of, in general, emerging synthetic biology technologies, but with specific focus on synthetic cells. We actually just had a paper in December in Science on one particular aspect of synthetic cell engineering, where we postulated that the risks outweigh the benefits, and we as international community are calling for international moratorium on doing this particular type of work. So this is an example of a possible response to the ethical and safety security considerations. We identify enough considerations or potential reasons to be concerned about the technology that now we're calling for not doing this work. So we're basically saying there is no safe way of doing it, so we should not be doing it. And this is just one area of the field. The way forward for the rest of the field would be to identify more pain points like that, to look at the technologies before they actually become a reality, identify what could potentially go wrong, what specifically could go wrong, and then what could be the safeguards against it. And if we cannot point out to good enough safeguards, then we should say, okay, we should not be doing it. Instead of waiting until the technology becomes a reality and then trying to safeguard it, we should be doing it proactively. And one of the ways of doing it is there was this so-called Asilomar meeting. It happened exactly 50 years ago. That was a meeting at the conference center at Monterey Bay called Asilomar, and that's why it became known as an Asilomar meeting, where people started developing frameworks for safeguarding synthetic biology. That was very early on in synthetic biology history. Now, on the 50th anniversary, we're going back to Asilomar actually next week. There's going to be literally the meeting that is going to be the Asilomar 2.0. And one of the 5 focus areas of that meeting is synthetic cell engineering. So we're going to try to repeat that, use that framework that the original Asilomar started, and develop the safety security analysis of, it's less ethics, it's more technical safety security of the development of different types of synthetic biology technologies. Another focus area for that meeting is AI and biotechnology. So that's something that a lot of you would probably find interesting, is how can AI kill everybody, and how can we prevent that from happening in the context of biological engineering?
Yeah, common themes. So actually, though, then maybe last question, but the possible power of this. The technology describing in Trumpet seem very much like the 3D biomanufacturing work where they use different DNA strands and they arrange themselves in 3D shapes and create effectively robots. They can then do computing much like you are describing.
Yes.
So how
Yes.
are these computational strands as well as 3D structural strands coming together?
They're coming together in this layering part. So we're using the 3D, we're actually working with someone who works on 3D bioprinting right now to build kind of a zones where different gates can perform different operations. So the gates is the kind of engine of it, and that 3D printing is building the chassis. So like that's the hardware, to use the analogy.
Yeah, that makes a lot of sense. And then you have the lattice of your cell, ultimately.
Yes.
Okay.
That's the end goal.
Yeah, well, fantastic. It sounds very promising, scary, in exactly the right mixture. Well, thank you very much.
Thank you so much, and thanks for all the questions.